Since the discovery of DNA's fundamental role in building and sustaining life, scientists have dreamed of having the ability to easily edit DNA in very precise ways. But why would they want to do this? Well, making specific changes to DNA sequences can help scientists better understand the function of certain genes, produce specific disease models, or even repair defective genes that cause diseases in humans. This is an exciting prospect, but methods to try and do this weren't practical or widely applicable. However, a few years ago, a gift from biology came from the basic research of the bacteria immune system, which gave scientists the ability to easily, customizably, and precisely edit genomes. Bacteria evolved ingenious ways of protecting themselves against pathogens such as viruses by using a system called CRISPR-Cas. CRISPRs are stretches of DNA sequence found in the bacterial genome. In close proximity to CRISPR are the Cas genes, which encode proteins necessary for the CRISPR system. Up until a few years ago, what was known about the CRISPR-Cas system is that bacteria infected by a virus incorporate elements of the virus's DNA into the CRISPR sequence. This protected the bacteria from future infection by this virus. Scientists observed that when a virus invades a bacterium, the CRISPR DNA produces one or two small RNAs called cRNA and tracer RNA. These RNAs bound to Cas proteins and formed complexes that cut the DNA of the invading virus, thus protecting the bacteria from infection. But many questions still remained. How did the small RNAs and Cas work together to detect and destroy viral DNA? In 2012, a group of scientists made a major breakthrough and discovered not only how the CRISPR RNAs and Cas cut DNA, but also how to create a new technique to specifically change the DNA sequence of any organism with great ease. This discovery came from a group of scientists led by Jennifer Doudna at UC Berkeley and Emmanuel Charpentier at Umeå University in Sweden. They published their results in Science in an article titled "A Programmable Dual RNA-Guided DNA Endonuclease in Adaptive Bacterial Immunity." So what exactly did these scientists find, and why is it so important for future biomedical research? First, the scientists dissected how Cas9 and the two RNAs can cut DNA. They found that the two RNAs, cRNA here in red and tracer RNA here in green, pair up, recruit Cas9 protein, and direct it to bind to the target DNA via the complementary base pairing between the red CRISPR RNA and the target DNA. Once at the proper DNA site, Cas9 cleaves both DNA strands. This cleavage occurs at a very specific and conserved position that is dictated by the sequence in the red CRISPR RNA molecule. This process is similar to finding your plane by matching the gate number to the one on your boarding pass. The scientists then wondered if they could engineer one RNA molecule that mimicked the structure of the CRISPR RNA and tracer RNAs bound together that would guide Cas9 to cut DNA at a specific location. The scientists designed one RNA molecule that consisted of the red and green RNA molecules connected together by a hairpin structure. In this case, they engineered the RNAs to target specific sequences of the gene encoding the green fluorescent protein, GFP. They added the engineered RNA molecules to the GFP DNA sequence, along with the Cas9 protein, and asked whether Cas9 would cut GFP DNA at specific sequences. And it did. When the RNA molecules were designed to bind to different regions of the GFP sequence, the GFP DNA sequence was cleaved at that specific location. On a gel that separates DNA according to size, you can see distinctly sized fragments of the GFP DNA molecule resulting from having been cut in a specific location. This was huge as it meant that scientists could engineer one RNA sequence, introduce Cas9, and cut DNA at a specific location of their choice. By having this simple and easily programmable system, scientists can now induce breaks in the DNA at precise locations. When the cell tries to repair the broken DNA strands by ligating them back together, it often causes a small insertion or deletion that changes the DNA sequence. scientists can take advantage of this process to add or remove specific DNA sequences at the site of the break. We now have a DNA word processor that can be used to change genome sequences, including our own. CRISPR has a bright future ahead to advance our understanding of human disease by creating a tool from basic research that is now widely used in the fields of molecular biology and genetics to change the genomes of any organism. But much work needs to be done to make the system more reliable. Use of the CRISPR system to edit human embryos is also a controversial issue, and researchers have already started the conversation about using this technology ethically and safely to advance human knowledge. This video has been provided to you by Eureka Science and iBiology, bringing the world's best biology to you.
It's getting late into January and the weather's getting crisper. I better put my coat on. A few days ago, a team of scientists from the Massachusetts General Hospital and Harvard research facilities announced a refinement to a genetic technique that may be a huge deal. Maybe the hugest deal. CRISPR isn't just the name of that drawer where your New Year's resolution to eat healthy goes to die. Clustered, regularly interspaced short palindromic repeats is a little bit of a mouthful, but it's actually a very descriptive name for what those crazy geneticists are up to. Let's review the basics. DNA is a chemical sequence of information that's built into a spiral, which you can find in just about any living thing. You can think of it like the design documents for how to make that thing, sort of like a bat or a mushroom blueprint. There's a lot of complicated chemical machinery to process DNA, but the basic principle is that some part of it gets translated into RNA, which you can think of as manufacturing drawings to produce some specific element of that design, which are then transcribed into proteins, which are the physical meat that make up things like cells and muscles and hair and whatever. There's an insane amount of complexity and nuance to that process. Like, which parts of the DNA get read depend on how it's physically contorted inside the cell. And if you start reading the same genetic sequence from here, you'll get a totally different set of proteins than if you started reading it here. The fact that with all those layers of complexity, we can still say something like, oh, she's got her father's eyes, is simply astonishing. Now, that's what's supposed to happen, and it happens in almost every living thing on Earth. But viruses have discovered a bit of a shortcut. Rather than replicating themselves, instead, they just put their DNA around someone else's machinery in the hopes that it'll get picked up and accidentally manufactured. Of course, proper cellular organisms don't want to go to all that effort to manufacture someone else's crap, so they've developed ways of identifying viruses and keeping their DNA from gumming up the works. CRISPR started off as one of those antiviral mechanisms. Bacteria that got sick of being infected by one particular kind of virus would actually keep that virus's DNA around inside their own, only with little brackets around it. The brackets are short palindromes of DNA code, like GAAT, TAAG, which let the rest of the cell know that it's not for fabrication. Quick side note, if you don't think that it's cool that life has a method for commenting out sections of genetic code just like any programming language, you can just get out of my face. Instead of making it, some bacteria use a protein that we call Cas, for CRISPR associated, which grabs a 20-letter segment of that code and then roams about the cell looking for a match. If it finds any DNA that matches, it cuts it. That cutting screws up any inserted viral DNA and prevents it from replicating. Cool, right? Not as cool as what comes next. If you're a geneticist who wants to tinker with certain sequences of DNA, a protein that locates and then does something to a specific sequence in a living cell sounds like a pretty good deal. Unfortunately, Cas tends to be a little indiscriminate in what it cuts. Which is a problem if you're a genetic engineer looking for something specific, because you can't really be sure what you'll get out the other side. Maybe the DNA will be snipped neatly just the way that you want, or maybe it'll look like a ransom letter. And that inaccuracy can be a huge problem. Gene therapy trials which showed remarkable promise have been stalled for years because, although they worked 90% of the time, the other 10, they gave people cancer. That's why this recent announcement is a huge deal. These scientists claim to have tweaked Cas9 to be extremely accurate, only touching the gene sequences that it's supposed to. Now, geneticists have a very precise, not to mention extraordinarily cheap and easy to use tool, which can locate, cut, deactivate, activate, or rewrite any sequence of DNA that they want in a living cell. If it works the way that they're suggesting it does, it's going to be like moving from a typewriter to word processing software for genetics. We didn't start tinkering with the nuts and bolts of genetics until the 70s, and historically it's been a pretty exclusive thing. You needed a specialized lab with hundreds of thousands of dollars of equipment and a staff of highly trained scientists to make the most basic changes. But with CRISPR, that exclusivity proviso goes out the window. Anyone can buy some Cas9 for a few $100. Any halfway decent lab can use it to alter the DNA of anything. We're talking about the level of access going from the smartest PhDs at the most prestigious research institutions to homebrewing in your garage. Now that anyone who knows some molecular biology can engineer any genome that they like, now that we can cheaply, easily, and accurately edit any DNA we care about, including human DNA, what's next? We can eliminate many monogenic diseases from the next generation of people if we want to. Just straight up get rid of them. Sickle cell anemia, cystic fibrosis, just not a thing anymore. It would be expensive, but it's certainly within our reach. We might be able to wipe out entire species on a whim. The biotech company Oxitec has engineered a genetically sterile mosquito that has decimated mosquito populations in Brazil. We might be able to do that to any species that we've decided we'd rather not deal with anymore. We can exponentially expand our knowledge of the human genome by quickly and easily producing new genotypes of mice to experiment with. Or we could start experimenting with the 20,000 known human genes to find out which combinations result in desirable outcomes. There's an established CRISPR methodology for every single one of them, ready to roll. And the thing is, with this update to CRISPR, all of these things have just become immediately relevant. This isn't some far-flung future thing. This is a few months from now. There's been a call by bioresearchers for a worldwide moratorium on genetically engineering humans that's been more or less respected. It was easy to keep an eye on the handful of labs that could do it, and it was a super complicated and very expensive process that wasn't well suited to a lot of practical applications. But now, every single person who's born with a genetic disorder that we've sequenced is going to have that moratorium to blame. There are a ton of ethical questions that come with CRISPR. I've talked about some of them before, but there's one in particular that's been bugging me. Now that we're at a point where we have the practical option to make future humans into whatever we want, what do we even want? For example, should our kids have the genetic makeup of super geniuses? I mean, intelligence is really useful and very powerful, but it is highly correlated with depression. What's more important, intellect or happiness? It was easy to philosophize haphazardly about this sort of thing when we didn't have the technology we needed to make it a reality. But there's nothing stopping us from determining the genetic basis of a real person's actual life now. And we have a lot of thinking to do about what that means. Please talk about it with someone, because we're going to need the answers in short order. And don't forget to leave a comment below to let me know what you think. Thank you very much for watching. Don't forget to blah blah subscribe blah share. And don't stop thunking.
I think the thing that's amazing about science and about biology for me is that there's always more to be discovered. And every time we feel like we've figured something out, it seems to open up several more questions. Scientists have appreciated for a long time that once we understood the DNA sequence in cells, if we had a tool that would allow easy manipulation of that sequence, that would be a very powerful kind of technology. CRISPR is a technology for changing the sequence of DNA in cells in a precise fashion to correct mutations that might otherwise cause disease. It's going to enable a lot of science to be done that was impossible to do in the past. So the way the CRISPR technology works is by the action of a protein called Cas9 that functions like a molecular scalpel for DNA. The CRISPR-Cas9 system has an amazing ability to recognize a particular DNA sequence in a cell that may be malfunctioning and then disable it by cutting the DNA. We call this gene editing, and we can use it to disable or repair a mutated part of the gene which may be causing disease. For Cas9 to find the malfunctioning DNA, we attach it to an RNA sequence that matches the DNA sequence we want to edit. Then we put this RNA-Cas9 combination into the cell, It finds the mutant DNA and uses a chemical reaction to cut the DNA strand right at the spot where it's malfunctioning. After that, we can sometimes insert the correct version of the gene for the cell to work properly again. It's a very exciting technology that's going to do a lot of good in human society and for human health. It stretches from human therapeutics to agricultural applications to thinking about how do we make better biofuels. These would be incredible outcomes, I think, of using this kind of technology in the future.
Imagine if we could stop mosquitoes from carrying malaria for good, or prevent ticks from transmitting Lyme disease, or eliminate the billions of dollars of damage caused by bugs to our food supply each year. Gene drives are a radical new approach to genetic engineering that could help us achieve these goals and a whole lot more besides. Yet, as you might expect, the technology is not risk-free. Gene drives are designed to eliminate unwanted traits in insects and other animals. They work by pushing out genetic modifications through whole species until eventually every critter has been changed into something we have intentionally engineered. The idea isn't especially new, but it's only very recently that advanced gene editing techniques have made human-designed gene drives possible. And at the heart of this revolution is a new technique for precision engineering genes, clustered regularly interspaced short palindromic repeats, or if you're not into brain-bending tongue twisters, CRISPR for short. CRISPR uses specially designed molecules that run along the strands of DNA in an organism's genome and seek out specific sequences of genetic code. Once found, they snip out the old code and paste in the new. It's essentially a sophisticated biological search and replace technology that allows scientists to easily modify the genetic traits of an organism, such as replacing the parts of a mosquito's genome that allows it to host malaria-causing parasites, for instance. Unfortunately, on its own, CRISPR isn't so effective. Every time a CRISPR mosquito mates with a wild one, its genetically modified DNA is diluted down, meaning that some of its offspring will still be able to carry the malaria parasite. And this is where the genius of CRISPR-enabled gene drives comes in. Imagine what would happen if that replaced sequence in the CRISPR mosquito also contained a little bit of code that continued to search and replace parasite-enabling DNA sequences. When the mosquito mated, the built-in code would ensure that every single one of its progeny would inherit the same traits, as well as inheriting the CRISPR code that would ensure the anti-malaria gene was passed on to every future generation. In other words, the new gene would be irresistibly driven through the whole mosquito population, and eventually every mosquito would become a human-designed malaria-free insect. And this is not a technology that's restricted to mosquitoes. Any organism that reproduces by sharing DNA through sexual intercourse is fair game. That said, designing an effective and responsible gene drive isn't a piece of cake. Working out which genes to change in an organism, for instance, is still an inexact science, especially when the CRISPR search and replace may modify more genetic sequences than expected or introduce unanticipated traits. The speed with which new genes can be driven through a species also depends on how frequently they reproduce. Gene drives are in principle great for transforming fast-breeding organisms like insects, yet it would take millennia to push a gene drive through a relatively slow-breeding species like humans, for instance. And that's assuming that naturally occurring genetic mutations don't mess up the recoding in the meantime. Nevertheless, gene drives do radically change how we're able to manipulate nature. And that also means they raise tough questions around risk and responsibility. For instance, we don't yet know how gene drives might harmfully disrupt ecosystems or create unexpected risks to other species, humans included. And we're not sure how much hardwiring in certain genetic traits will weaken an organism's ability to adapt to changing environments, especially when those traits would normally be rejected through natural selection. And then there's the danger of entrepreneurs and hobbyists even playing around with nature just because they can. Or people who intentionally set out to create even more dangerous species, weaponized super mosquitoes possibly, or Hunger Games-like species designed to maim and kill. Not to mention the possibility of groups starting exclusive human gene drives that will give their future heirs genetically enhanced abilities. Because of concerns like these, scientists have already started to think about how gene drives might get out of hand. Recommendations are beginning to emerge on responsible practices like testing gene drives in the lab before trying them out in the environment. And there's talk of countermeasures such as follow-on gene drives that can reverse changes or block the propagation of genetic modifications. Yet these discussions often assume that it's the scientists and the developers who know what's best. I suspect, though, that when it comes to intentionally redesigning natural species, there are a lot more people who'd like a say in what should and should not happen. Especially as the risks and benefits of gene drives are likely to affect far more people than those who came up with the bright idea in the first place. Which leaves the question, if the use of gene drives are a social issue as much as a scientific one, how can ordinary people become involved in ensuring we get it right before it's too late? If you have any thoughts on this, please do share them in the comments below. And as always, stay safe.
I have thought a lot about the way that the CRISPR technology might be used, and I mostly feel excited because I think that it's a very exciting technology that's going to do a lot of good in human society and for human health. I do think that there are reasons to be cautious in the way we implement it for various things, in particular for doing clinical applications in embryos, like human embryos. If you do the editing in adult tissues, those are changes that are made in an individual, but they're not passed on to children. I think most people would feel that genome editing in adults is, or at least might be, for some applications, a very appropriate technology. It might be analogous to taking a pill or any other kind of therapeutic drug that you might use in a patient for treating cancer or some other disease. And if you could do something profound like eliminate sickle cell anemia or eliminate cystic fibrosis, or have someone not have to suffer from Huntington's disease anymore or worry about getting it when they get older. What certainly raises ethical implications is whether that sort of change should be made in eggs or sperm or embryos, where those changes could then be passed on to the next generation. So I feel that it's appropriate with guidelines to conduct research in those kinds of systems, even in human embryos. I don't think that it would be appropriate to use this technology, at least today, for any kind of clinical application in human embryos. The science is going 1,000 miles an hour. One of the roles that we as scientists need to play is to really communicate the power of this technology and how we can be responsible in using it. For that conversation to carry weight globally, it really needs to be a conversation that involves scientists that are international. Do we know enough about the human genome to understand the impact of making changes to it in a developing embryo? And so I think that there I would like to see our society draw a line and say that, you know, we won't go there right now. We'll take the time that we need to really think about this.
Bacteria may hold the key to fighting off certain viruses in patients once and for all. In a new study published in PLOS Pathogens, researchers in the Netherlands used the hot new genome editing system known as CRISPR-Cas to target herpes viruses, some of the most common and persistent viruses that infect people. The technique, which is based on a bacterial defense mechanism, works in human cells to limit replication of the herpes virus and in some cases, even allows for their complete eradication. Herpes viruses are all around us. The family includes the herpes simplex viruses that cause cold sores, recurrent eye infections, and genital herpes, as well as others that can cause birth defects, spread mononucleosis, or cause cancer. Nearly all adults are infected with at least one of them, but many people probably don't know it. That's because the viruses are very good at staying under the radar and out of the way of our immune systems. During flare-ups, the viruses pop out more copies of themselves and can be treated with existing antivirals, but those drugs can't eliminate the virus. The infection is lifelong. The CRISPR-Cas system offers one way to seek and destroy these latent viruses. Short guide RNAs can direct the Cas9 protein to specific viral sequences, allowing it to break the DNA at precise locations. This editing is so damaging that it kills the virus. If it can reach all existing copies, it's a potential cure. The Dutch researchers first tested whether CRISPR could work against Epstein-Barr virus, which causes mononucleosis and drives certain cancers. They designed guide RNAs to target several critical viral genes. In a model of latent EBV infection, they found that single guide RNAs, together with the Cas protein, successfully removed the virus in 40 to 60% of cells. Using two guide RNAs sequentially worked even better, leaving more than 95% of cells virus-free. Next, the scientists turned to human cytomegalovirus, or HCMV. It's responsible for birth defects and can be deadly to people with weak immune systems. CRISPR successfully limited viral replication, but it was important to choose the right targets. Guide RNAs against inessential genes failed to slow viral reproduction, and different strains of HCMV were more susceptible to some guide RNAs than others. In human cells infected with herpes simplex virus type 1, CRISPR also blocked viral replication. In contrast to HCMV, even when the researchers targeted non-essential viral genes, it still worked, although to a lesser extent. Using two guide RNAs again proved to be most successful, in part because two targets make it harder for the virus to mutate and escape treatment. While virus titers from infected cells fell by as much as 10,000 times with a single guide RNA, viruses were undetectable in dual-treated cells. CRISPR could also block replication of reactivated virus, but couldn't edit latent virus. HSV1 viral genomes persisted despite treatment. The results suggest that CRISPR is an effective way to limit replication of multiple herpes viruses, but the holy grail, eradication, so far is only possible for Epstein-Barr virus. One reason for this difference might be the availability of viral DNA to the CRISPR system. EBV's genome is located in easily accessible circular molecules within the nucleus of dividing cells, whereas HSV1's genome is closed off in non-replicating neurons. Future work may reveal a way to target latent genomes for HSV1 as well. Until then, anti-herpes virus CRISPRs have several promising applications that await testing in animal models or humans. They might remove cancer-causing EBV from tumor cells, help rid donated organs for immunocompromised patients of viral invaders, or prevent cold sore outbreaks and recurrent eye infections by blocking HSV1 reactivation. All thanks to the defense mechanism of bacteria.
Our genome is a huge book of billions of letters, A, T, C, and G. So those sequence of the letters are the genes we have. Most of the time, a disease happens not because of one gene have defects or multiple genes. So we need to know how to modify multiple genes simultaneously to be able to cure disease and to study biology. A couple years ago, it's very difficult to even make one modification in one locus. But now, I think it's become possible to make multiple changes in the genome. When you want to make a change in the genome, you really just need to go there and design molecular scissors to make a cut. And then you can have all kind of magic you can play with there. There are now three major classes of tools becoming available, like zinc finger nucleases, TALEN, and CRISPR. The first two enzymes are literally like a scissor. So they have two halves, and each half recognizing part of the DNA sequences, they come together and make the cut. And the CRISPR is different because now your protein, Cas9, doesn't know where to cut, but it complexes with this small RNA molecule. And the RNA, if you know biochemistry, is made by 4 different flavor nucleotide, slightly different from DNA. So they can naturally pairing with the DNA. Now you can have this perfect small molecule that targeting wherever in the genome you want by base pairing and guide the Cas9 protein to go there and make the break. So when this system was defined, the first indication to us is it's a perfect tool for multiplex genome editing because you can express one protein very large, but many small molecule of RNA very easily. So we showed that in a single cell, you can efficiently kill 5 genes simultaneously in a single cell, so which is quite remarkable at that time. And then you can also introduce the system using a fine needle to inject them into the fertilized egg, which is the first cell of our life. So this is a mouse embryo, a mouse zygote. So when you inject the CRISPR system in there, they start to cut the DNA at the very beginning of life. Once you modify this cell will develop into a whole mouse. So the mouse will have the exact same mutation in every single cell. So by doing that, we show you can actually introduce precise modification in multiple genes in one step. You can also insert in large piece of sequences in defined locus in a multiplexed way. You generate an animal in one step. And this principle can be applied to other species as well. In this example, each little ball is actually a mouse embryos developed 4 days after your one cell injection. And we put in different color protein after the endogenous gene that's expressed in those stages. So now you can see these 2 genes are labeled with 2 different colors, showing we can actually inserting thousands of nucleotide sequences in the defined locus. If you do this simultaneously, you can get orange color embryos, means you can simultaneously introducing 2 large pieces of DNA into 2 unique sequence in the whole organism. So this is, I think, very fascinating because it dramatically reduced the cost of making genetic modified animal as well as reduce time frame you need to make those animals, comparing to the traditional method of gene targeting, which was awarded Nobel Prize for that in 2007. We believe this will actually becoming the new standards of making animal models. However, there are obviously ethical hurdles we have to cross to apply this to human. Now our focus is really to apply this to human in the somatic cells. So our favorite cells is the T cells. The left side is red blood cells and the red blue cells are the T cells. One of the most important effector cells for immunity. Why we want to do this and why we want to do this in a multiplex way? Now, it's a very simple cartoon to show how this works. So the T cells express a protein in their surface called T cell receptor. And this receptor will recognize another cell's HLA molecule. If they think this HLA is from another individual, they will kill the cells. And if this HLA complex with some pathogen antigen, they will also kill the cells. So now you imagine, if I donate my T cells to you to combat your disease, it wouldn't work very well because my T cells have TCR that are going to kill your normal cells because we think it's alien. But you also have a lot of T cells. Your T cells will recognize my T cells as alien. So you're going to kill my T cells. So my T cell would not last long and will not be able to do it very well. So if you kill both TCR and HLA on my T cells, this becoming a cell I can donate to you, and that's supposed to work much better. But we still need to prove this. With the power to really engineer the genome with great ease, also in a multiplex manner, I think the question we're asking is really, how can we utilize this? Many possibilities we don't know before. And to what extent should we modify human cells? Because I think it's a question about somatic cells, other question about the germline or the human embryos. But I think the line is very blurry now because a lot of stem cell biologists are making the magic of turning every single cell, somatic cells, into a pluripotent stem cells or into a germ cells maybe. Even if I modified your fibroblasts or skin cells, potentially in the next 10 years or 5 years, you can turn them into a sperm. So where is the line? I think it's a very interesting time we are now. Thank you.